Now in this portion we will be discussing the process of urine formation. The whole process starts right here in the renal corpuscle, where there will be filtration into that capsule. And then from the capsule, what is now known as the filtrate is going to go through the proximal convoluted tubules down to the nephron loop, the descending followed by the ascending limb, and then the distal convoluted tubules, and then to the collecting duct. You can see that the collecting duct gets urine from several different distal tubules, and then all of that urine will be dumped into the minor calyx. From the minor calyx down to the major calyx, then to the renal pelvis, down the ureter, to be stored in the bladder, and then when we are ready to eliminate urine, it will be eliminated through the urethra. You'll see here that the process of urine formation really ends at the collecting duct. So anything beyond the collecting duct, all of these structures are not going to change anything in the composition of urine. So the actual formation of urine involves these structures, while starting at the minor calyx all the way through the urethra, these are really there for urine excretion only. Now let's talk about the 3 steps of forming urine. The very first step is glomerular filtration, followed by tubular reabsorption and tubular secretion. So we'll talk about the process looking at the capillaries and the tubular system. You can see here the afferent arteriole bringing blood into the glomerulus and then exiting through the efferent arteriole, and these are the peritubular capillaries. The very first filtration process happens here in the glomerulus, where everything except for the big plasma proteins are going to filter out into the capsule. Now that filtered urine or filtered fluid is now known as the filtrate. The filtrate passes through the proximal, nephron loop, and distal tubules, and it has to change in its composition. It is not urine yet. So there is one step known as tubular reabsorption, where things that are important to the body that have been filtered out, for example, glucose, amino acids, some ions, the body needs them. So these these molecules are going to be reabsorbed into the peritubular capillaries. So tubular reabsorption is the passage of important molecules from the renal tubules into the peritubular capillaries. Well, tubular secretion, on the other hand, is the opposite. Things that have not been filtered and the body would like to get rid of. So tubular secretion would be the elimination of molecules from the peritubular capillaries and secreting them into the tubular fluid. Now all of that process, all 3 of these steps, filtration, reabsorption, and secretion, end up making urine. First step again is glomerular filtration, where everything that's in blood will be filtered with the exception of the big molecules of plasma protein. So the filtrate really ends up having the exact composition as interstitial fluid. And we've seen the structure of the glomerulus, and we've seen the podocytes surrounding the glomerular capillaries. The glomerular capillaries have very big pores compared to the capillaries of the rest of the body, and that's because in order to accommodate their function of filtration, these capillaries are surrounded by all of these podocytes with the little slits in between. And again, that is going to assist in filtration but prevent the molecules that are too big to pass out. We'll keep those bigger plasma proteins inside of the capillaries and filter plasma with all of the small molecules into the glomerular capsule. Now, what is the pressure, or what are the pressures that play a role in filtration? So there are several pressures here. Blood passing through the capillaries leads to a hydrostatic pressure, and that hydrostatic pressure in the glomerular capillaries is known as the glomerular capillary hydrostatic pressure. Well, the capsule surrounding the capillaries also has water in it, so it also has its own hydrostatic pressure, and that is known as the capsular hydrostatic pressure. There is a third pressure, and that would be due to the plasma proteins that are found in the capillaries, and that pressure is known as the glomerular capillary osmotic pressure. Out of all three of these pressures, there is only one of them that helps in filtration, and that is the glomerular capillary hydrostatic pressure. So the hydrostatic pressure here in the capillaries is the only one that helps or favors filtration, while the other two pressures, which are the pulling of the due to the plasma proteins and the hydrostatic pressure out here in the capsule, both of these oppose filtration. But the net result of all three of these pressures end up favoring filtration. Here you can see how much we filter per day. So about 180 liters are filtered every single day, but we really only urinate about 0.6 to 2.5 liters of urine per day. So the kidneys are going to reabsorb all of that filtrate back. The glomerular filtration rate is how much was filtered per any given time unit. So you see here that in an adult, healthy average adult filters about 180 liters of filtrate every single day. The blood plasma is filtered about 60 times per day. But again, only a small percentage of all of that filtrate is going to be eliminated as urine. So where does all of that go? Again, remember the process of tubular reabsorption. We are going to reabsorb all of that water that the body still needs, along with the glucose and amino acids and the important molecules through, again, that second step, which is tubular reabsorption. Now, how does the kidney control its own filtration rate? Where there are several processes. The most important is autoregulation, where the kidneys, through these sympathetic impulses, can change the diameters of the afferent and the efferent arterioles. There are two other mechanisms, for example, the renin-angiotensin system, which we'll discuss in more detail in the next slide. And there are other hormones like the atrial natriuretic peptide, which is a peptide hormone secreted by the atria of the heart in response to high blood pressure. And what it does is that, according to its name, it dumps the sodium in urine. When you dump sodium in urine, sodium through its osmotic pressure is going to pull water with it. So you are now excreting more sodium and water, and that decreases the blood pressure. Another hormone is known as the ventricular natriuretic peptide. The B stands for the ventricles of the brain. So the ventricular natriuretic peptide is a peptide hormone made by the brain ventricles, not the ventricles of the heart. And again, it is secreted in response to high blood pressure, and it leads to the increase of sodium excretion and water excretion. I want to get to this autoregulation here for a minute before we talk about the renin-angiotensin system. So I'm going to go back a couple of slides and take a look at how the sympathetic nervous system can regulate filtration by changing the diameters of these blood vessels. So usually the afferent arteriole has a bigger diameter than the efferent, which means that more blood is going in than is leaving. And that leads to a higher hydrostatic pressure and helps in the filtration process. So I want you guys to think of a couple of scenarios. What if the afferent arteriole dilates, that is going to lead to more blood coming in to the glomerulus, which increases its hydrostatic pressure, which increases filtration. Well, what if the afferent arteriole constricts? Well, that constriction leads to decrease in the hydrostatic pressure of the capillaries, which decreases filtration. Now I'll give you one more scenario, which is the constriction of the efferent arteriole. When you constrict the efferent arteriole, that means the hydrostatic pressure in the capillaries increases, which also increases filtration. Now you think of the very last possible scenario, which is dilation of the efferent arteriole, and hopefully you are able to figure out how that would affect the filtration process. Email me with the scenario that you thought of so that we could check it together. So that is the whole process of autoregulation. So again, you want to be comfortable with those 4 different scenarios: the constriction and dilation of both the afferent and the efferent arterioles, and how that would affect the filtration process. Now, the renin-angiotensin system starts by a protein made by the liver known as angiotensinogen, which means that it is an inactive protein because it needs to be activated, and we know that because it's telling us that it can create something. So this inactive plasma protein known as angiotensinogen, synthesized by the liver, is secreting or is circulating in the plasma in a scenario where there is low blood pressure. And that decrease in blood pressure is detected by the juxtamedullary apparatus. That will stimulate the kidney to release renin. That hormone renin, now in the bloodstream, will activate angiotensinogen into angiotensin 1. Angiotensin 1 needs to be activated in a second step through an enzyme known as ACE, or angiotensin converting enzyme, into angiotensin 2. Now, angiotensin 2 will do anything it can to bring blood pressure up back to normal. So it'll make us thirsty, so it increases thirst, it vasoconstricts, it increases the levels of 2 different hormones, aldosterone and antidiuretic hormones. Basically, what those 2 hormones are going to do would be conserving sodium and water and preventing them from being lost in urine. Now that is going to take us to our next process of secretion, tubular secretion and tubular reabsorption, which I am going to leave to the next tutorial.